Good morning, everyone. Welcome back to our channel. Today we are here to discuss important MCQs on cytoskeleton. So, this is the cytoskeleton. This structure involves microtubules, intermediate filaments, and microfilaments. This provides the shape to the cell. So, here we'll discuss some of the important MCQs regarding cytoskeleton. Coming to the first question, the identification of actin filaments in a cell can be done by using the protein. Which protein here? Option A, myosin. Option B, kinases. Option C, globulin. Option D, vimentin. And correct answer is myosin. Coming to the actin filament, the protein here involved is myosin. Coming to the microfilament, the protein is the kinesin and dynein. Two proteins are involved in the microfilaments. So here correct answer is for actin filament is myosin. Question number 2. Microfilaments are mostly involved in which process? Option A, activation. Option B, stimulation. Option D, degradation. Option D, motility. And here correct answer is motility. It involved in movement, cellular motility or cellular movements such as movements of vesicles, phagocytosis and cytokinesis. Question number 3. Which statement is true regarding microtubule or microfilament? Option A. Microtubules are composed of tubulin. Microfilaments are composed of actin. Intermediate filaments are resistant to cytoskeleton B and colchicine. These are the two toxins that inhibits the functions of microtubules and microfilaments. So here the correct answer is all the above. Intermediate filaments are resistant to these toxins. Microtubules are composed of tubulin. It contains two tubulin monomers, that is alpha and beta tubulin. And microfilaments are composed of actin filaments. The correct answer is all the above. Question number 4. Microfilaments are composed of globular subunits of which protein? Here microfilaments composed of the actin. Here the correct answer is actin. So myosin, it is a motor protein. It is also kinetic as also motor protein. Colchicine is a toxin produced from the plant-based toxin. Actually, it is a drug used for the control of the cancer. Here, how it correlates means for cell division, the actin and everything it requires, it polymerizes and depolymerizes the actin filaments so that re- restricts the cell division. That finally that controls the cancer. Question number 5. The drug cytoskeleton blocks the option A microfilaments, option B pulse plus end of microfilaments, option C minus ends of the microfilaments, option D none of the above. And here this drug in blocks the plus end of microfilament. Here this is the microfilament. It contains both positive end and negative end. This drug blocks the positive end of the microfilament. Here, if this is the cell, in centromere the positive ends minus and negative ends cross this way. At the centrosome, negative strand, negative end will be there at the towards the and positive strand goes towards the cell wall plus or plus one number. Question number 6. Electrophilin blocks which among the following? It is the drug produced when the sponges. Option A, plus end of microfilaments. Option B, minus end of the microfilaments. Option C, actin monomers. Option D, actin filaments. It blocks the actin monomers. This actin monomers forms the actin filaments by polymerization. It blocks the actin monomer so that the actin filaments don't form and it depolymerizes the and polymerizes. Question number 7. A network of microfilaments and microtubules is classified as option A cytoskeleton, option B active skeleton, option C vertebral skeleton, option D endoplasmic skeleton. Here already we know that answer is cytoskeleton. This is the cytoskeleton. It includes the both microfilaments and microtubules. You can see here microfilaments, microtubules. Here's the structure to the cell. So we'll call it as the cytoskeleton. Question number 8. Which type of myosin protein move towards the minus end of the microfilament? Option A myosin 1, option B myosin 2, option C myosin 4, option C myosin 6. Here this myosin 6 move towards the minus end. This is the cell here centros. So here minus end will be there, here positive hand. Here myosin protein moves towards the negative end or minus end, that is myosin 6. Remaining all myosin proteins move towards the positive end. Question number 9. The head of the myosin binds to which filament? We know it actually. Option A ATP, option B ADP, option C actin filament, option C neurofilament. Here myosin protein, this is for actin filament. Here myosin protein head actually binds to the actin filament and it moves. This will call it as the motor protein. It moves from the along with the actin filament. Question number 10. Actin filament serves as a trace for the movement of which motor protein? Option A myosin, option B kinesin, option D dynein, option D none of the above. Here option is actin filaments in acts as a track for the movement of motor protein, that is the myosin motor protein. These two are microtubulins. For these two motor proteins, microtubulins acts as a track for the movement of these two proteins. Thank you for the listening. And here answer is